Hello. So today we'll discuss about DNA replication in eukaryotes. So here in in eukaryotes, here we have DNA polymerase alpha, beta, gamma, delta and epsilon naught. So here, because of the complexities of chromatin structure, eukaryotic replication moves at around 50 base pair per second. Right. So in eukaryotes, clusters of about 20 to 50 replicons initiate simultaneously at defined times throughout S phase. So centromeric and telomeric DNA replicates last. Okay, so now starting with DNA, so here it replicates 3000 base pairs per minute. Now here we have DNA polymerase alpha, beta, gamma, delta and epsilon. It's epsilon. Okay, so now it's in eukaryotes, whereas in prokaryotic replication it was DNA polymerase 1, 2, 3, whereas in eukaryotes we have DNA polymerase alpha. So now here we have two stages or two steps. So first is pre-initiation complex and second is formation of initiation complex. So now in pre-initiation and initiation complex, here we have some CDKs. Here is the role of CDKs. CDKs are cyclin dependent kinases. So what happens if CDK concentration is low? What will happen? It formation of pre-initiation complex. Now if CDK concentration is high, now how this works is we have two phases, G1 and S phase. G1 is the preparatory phase. Here its preparation start. G1 is the preparatory phase and S phase is the here occurs the DNA replication. So when CDK concentration is low, now this is occurring in G1 phase. So in G1 pre-initiation complex is formed, and on the other hand we have activation of existing initiation complex. Now when CDK concentration is high, what happens? It will activate the existing initiation complex. Existing means previously, like earlier initiation complex that was formed, it will activate it. So now when concentration is high, that means it will come in S phase. So in low, formation will be favored and activation will be inhibited, means activation will not be there. Similarly, when concentration will be high, activation will be favored and formation will be inhibited, means this will not be formed. Okay, now here let's start with the DNA. This is its origin. Now here we have a protein. We have some proteins, DNA polymerase, RPA acts like SSB. Then RFC is there. RFC is a clamp loader. ORC is origin recognition complex. Then MCM is there. Then CDK is there. So now here we'll talk about the protein ORC. Now origin recognition complex, it will recognize this origin. ORC, origin recognition complex. Now what happens after it will recognize its origin? It will recruit other proteins. It will recruit CDC6 and CDT1. So basically this CD6 and CDT1, these are helicase loaders. Helicase loader means it will help to recruit helicase. So now once helicase loaders are recruited, we have another protein, that is MCM27 complex. This is helicase only. In eukaryotes we usually call it MCM. So in eukaryotes MCM name is given instead of helicase. So now this CDC, it will recruit MCM. Okay, so this was all happening in G1 phase because CDK concentration is low here. Now what will happen next? Now here forming a replication bubble. Okay, now coming to DNA CDK concentration. Now here what happens, CDK concentration increases. One sec. I'll write this later. Okay, now coming to DNA polymerase alpha. Now we have here we have DNA polymerase alpha. Now its job is, it acts like a primase, like it adds primer. Now here primer is added, so obviously this will be 5' end, this will 3'. So now primer is added, and it along with that here it will add first nucleotide, adds first nucleotide, and it adds 20 nucleotides. So here firstly this is this white color is primer. Now 20 nucleotides are added by DNA pol alpha. So similarly here also since this is a lagging strand, now this is the leading strand and this is the lagging strand. Now here extension will be done by DNA pol alpha. Now what happens here, we have discussed DNA polymerase alpha. Now we have DNA polymerase beta, gamma, delta. So now DNA polymerase delta, it is responsible for lagging strand synthesis, and epsilon, it is responsible for leading strand synthesis. Since this is the leading strand, here comes the epsilon, and this is the lagging strand, so here comes the delta, DNA delta. So further extension will be done by polymerase alpha, but epsilon and delta, they have a role in leading and lagging strand. Okay, now coming to, and we have another protein, that is RFC. RFC, this is a clamp loader. In prokaryotes also there was a clamp. Similarly in eukaryotes, clamp is RFC. RFC is replication factor C. So now what happens? RFC loads pCNA ring. pCNA ring, it's like clamp. Now here so now RFC, replication factor C. Now what's the role of RFC? It will recognize junction between primer and nucleotide. Here are, this is the junction, right? This is the primer, this is the nucleotide. So it will recognize this junction, RFC. Primer and nucleotides. So pCNA ring, this is like a clamp, and this is for this delta and epsilon strands. So now here CDK activity increases. Now when CDK activity increases, that means it is all occurring in S phase. So here we have helicase loader, CDC. So role of helicase loader, it will load helicase, this helicase. Now function of helicase is, it will separate strands, it will unwind strands, the origin will melt. Okay, now this whole machinery, one sec. Now here one more thing is there. We have another protein, that is CDC45. MCM gens. So this CDC45 is also recruited here. Okay, so here CDC45. So at the beginning of S phase, MCM phosphorylation by DDK, that is DBF4 dependent kinase. It promotes the recruitment of CDC45 to MCM ring. Beginning of S phase means when CDK concentration is high. So it will, when CDK concentration is high, it will result, it will recruit CDC45, that is MCM and GINS. So this is done at the beginning of S phase. So S phase means when CDK concentration is high. Okay, so now this whole machinery replicates and further forms the process. So this was about initiation in eukaryotic replication. Now let's see the difference. Now in prokaryotes, if you remember, it was DNA polymerase 1, 2, 3, right? So the main enzyme was DNA polymerase 3. It helps in synthesis of leading as well as lagging strands, right? So then DNA polymerase 1, its function was it removes RNA primer. Now DNA polymerase 2, it helps in DNA repair, and it has 3' to 5' exonuclease activity. Whereas if I see eukaryotic replication, we have DNA polymerase alpha. Alpha acts as primase, adds primer, and it adds first nucleotide. It adds 20 nucleotides, right? Now coming to DNA polymerase beta, its role is in DNA repair. Alpha, beta, then gamma. Its role in it does DNA replication in mitochondrial DNA. Alpha, beta, gamma, then delta. It is responsible for synthesis of lagging strand. Epsilon, it is responsible for synthesis of leading strand. Whereas in prokaryotes, DNA pol 3 synthesizes both strand leading as well as lagging. In eukaryotes, we have different polymerase for leading and lagging. So then in prokaryotes, the length of Okazaki fragments is more. Length of Okazaki fragments is more, whereas in eukaryotic replication, the length of Okazaki fragments is less. Then in prokaryotic replication, because it has only it has single ORI, right? So it forms single replication bubble. Only one replication bubble is there, whereas in eukaryotes we have multiple ORI origin sites, origin regions. So here we have like this, then this, then this. Here we have multiple replication bubbles. Okay, so this was about it. Initiation. We have two more stages, elongation and termination, that we'll discuss later. Okay, thank you.